You need to bring lots of diversity from these wild strains into the commercial strains, and this genomic technologies. We've got the muscle here to throw these at kind of lots of the world's problems, and so they've been at the forefront of trying to adapt it and use it in other areas of research, trying to kind of like genomicsify them. So they have a team that's been doing a lot of stuff on sort of almost quantitative biodiversity research, where looking at environmental DNA or even bulk. They do bulk DNA. They call it genome sweeping. For things like, so basically getting bees and getting the pollen from bees and basically studying and figuring out the biodiversity of all of the flowers in this region. What are the bees eating? What flowers are growing here? Bulk sampling of insects from the traditional biodiversity. You'd set up a little trap with a light and collect a load of moths or whatever, but they can actually sample of this quantitatively, basically sequencing the goo from like those squished up insects and seeing how sampling huge amounts of sampling sites, huge amounts of data, and also quantitative. There's more butterflies here than wasps, and this. It's quantitative. You can actually see this from spiders' webs. You can actually see what the spiders have been eating from the DNA in the webs, or stream water. You can figure out what species live in the stream, even if you've never even seen them. So these are, DNA sequencing has got so cheap and it's so sensitive that these are the applications that it's kind of moving to.
But the dinoflagellates could be, very useful in that, because if they're having this like super complex.
So that they're very, very strange. The genomes can be up to almost 100 times larger than the human genome, which for a unicellular organism, we're pretty biased to we're very biased to humans. We think we're the most complex thing there is, but one of these tiny dinoflagellates is like technically can be up to 100 times more complex genome than ours. And so if it took us 20 years and $3 billion or whatever to sequence the human genome, there's been an incredible drop in the price of sequencing, but still we've got up to sequencing some of these tree genomes with. They're about 10 times bigger than humans, or amphibians are incredibly complicated. They just completed the axolotl, which is about 10 times bigger than a human. But that's basically the limit of where we're at, and so dinoflagellates are still we're still a decade or two away from actually being able to properly look at the genome of these.
But is the complexity of the genome also dependent or in relation to its place in evolution? If now you have the amphibians and the dinoflagellates being so complex, because they might have tons of information about how evolution.
I don't know. There's a lot of arguments over this genome size issue, like what it actually means. Things have got they're old. These are very old organisms. So over time, you can accumulate lots of things. But then some things some are theoretically, old things and are not complex. I don't know. You get duplications because of viruses. You get thing for various. So in plants, that they've seen that the genome duplications actually have been used in evolution to create new genes and new. There's been they can kind of see evolutionary reasons why this has been useful in plants diversifying and changing. But first, when it gets to that level of complexity, it's BGI has a big agriculture arm. They are working with Gates Foundation, Mars Seeds of Change, various organizations looking at orphan crops, food security. With climate change, very rapidly, agriculture has to adapt to the grain belts moving, the rice belts moving, where all of the world's food are. Soils are losing their fertility, pollution, salinity, things like this. And so BGI has been kind of using a data-driven approach to this, basically throwing their sequencing muscle at looking at all of the genetic diversity of lots of these species, particularly crops that have been studied. All of the crops that people in India and Africa use, they're not profitable for the big seed companies in Europe and the US, and so the amount of effort in breeding and improving these crops has been minimal because it's not profitable, but it feeds so much of the world, and so this is why a lot of these foundations have been funding research, and BGI has been providing them the data for this research to improve these crops, tailor them very, very rapidly. So they're not GMO. This is data. This is genome assisted, genome data driven. Historically when you breed plants, a lot of it's guesswork. This is taller than this one, so you assume this one is the one you want. But there's useful genes, useful mutations, and not useful mutations. Maybe the taller one isn't the one you want. It's this other one here, but you can't tell that from physically looking at. But from this genetic information, it's very, very quickly breed sequence. They developed these breeding platforms to massively increase yield of millet was the first one, but they're doing it in lots of other crops, because millet really can grow in these really resource poor environments. It's a kind of poor person's food in the north of China and Africa and places like that. So they're really focused on improving that, and so that's a kind of agricultural work. So BGI it's got an interesting history. It was founded in the anniversary was yesterday actually, 9/9/99, and because 9 is it's like a lucky number, longevity, I think, and so they came up with the Human Genome Project. They wanted China. The founders were sea turtles. They were PhD students, postdocs who studied in the US, in Europe, and were at Washington University when the Human Genome Project was starting. And they came back to China and were pitching to everybody, "Yeah, we need to do this, and China needs to participate." And they were just turned down. People said, everybody, all the funders, all of the policy people, all of their kind of peers were just like, "Why do you want to work in this?" Either the public project will win and everything's going to be free, or the commercial project is going to patent everything, so we wasted all our money, so why participate? So they wouldn't take no for an answer, and basically got loans. The Sanger Institute just lent them a machine. One of the founders had a small company that he sold and used as kind of seed funds, and they did 1% of the human genome. And then when Bill Clinton and Tony Blair were announcing that the Human Genome Project had been completed in 2001, they were saying, and Japan has done 7%, and Germany has done this amount, and China's done 1%. And that was the first time that the Chinese government heard this and were like, "What? We've done 1% of the Human Genome Project. How did that happen?" And then basically kind of felt guilty, and then paid the BGI founders back for doing this. And so that was basically BGI's startup funds. And then they did the rice genome, the first rice genome. they then went to the big consortium in Japan, and they said, "Look, can we join this?" And they apparently the founder says that he turned up to a meeting and he was stood up. They didn't even like see him because they're just like, "Who are these Chinese people?" He turned up with flowers and there was like nobody there. And so they went back to China with no money. The staff were handing around baskets and they were paying for sequencing reagents with like the employees' donations and stuff. And they basically sequenced the rice genome first, and like embarrassed Japan and embarrassed all of these people. And it was a Monsanto and Syngenta did a genome at the same time, private. You had to subscribe to access the data and stuff. So BGI released their one openly, killed the business model of the Monsanto Syngenta one because it was open.
I didn't know that part of it.
Yeah.
Yes, no.
And so basically they've just been like troublemakers basically for nearly 20 years. Human rights. They then were in the sort of Chinese system a bit in Beijing, and then left in 2006, 2007. Came to Shenzhen because they kind of fell out with Chinese Academy, and there was a lot of bad blood with Beijing, and so moved here and moved to like Shenzhen side of the border. Got lots of money from the Shenzhen government to come, and basically everything's grown from that. When they came to Shenzhen, there was like 500 people, and now there's 7,000. So
because the demand is growing, and also the machines are.
Yeah. Technologies. No area of research has like grown at this scale. Like improvements in output and scale, it's outpaces Moore's law. The computers are not growing as fast as the sequencing output, as it like improvements in sequencing output. So it's getting harder and harder to crunch it. So for like a big project like the rice genome, they did a 3,000 rice genomes and made like the raw data was about 15 terabytes, and then the computers here couldn't crunch that amount of data, so they had to take it to Guangzhou. Tianhe supercomputers, Tianhe 2 is the world's largest supercomputer. They just crunched it on that, and then the process data was 100 terabytes, so they got back on like all of these disks. And but this is the scale of the stuff that they're doing.
Wow.